Hi, my name is carola.greve, and the title of my poster is Providing Solar-Powered Sea Slugs with High-Quality Genomes. So, what are solar-powered sea slugs? These slugs belong to a small group called Saccoglossa, which comprise about 400 described species. They are able to incorporate and maintain the chloroplasts from their food algae in their digestive gland cells. Inside the slugs, the chloroplasts still do photosynthesis, producing starch, and it is assumed that these chloroplasts help these slugs somehow to survive weeks or months of starvation. This phenomenon is called functional kleptoplasty. So how keep the slugs, the chloroplasts, active? No one knows. This is why we would like to compare the genomes of Elysia timida with the already published genomes of Placobranchus ocellatus and Elysia chlorotica. These three slugs are all able to incorporate the chloroplasts for at least 6 months. For the genome of Elysia timida, we extracted the DNA from a whole individual. We prepared a PacBio ultra-low DNA input library, and this library includes a whole genome amplification step. In the first try, we also prepared a PacBio low DNA input library. And as you can see, the sequencing results are quite surprising, because sequencing the low input library gave really bad sequencing results, and the output was only 10 gigabases. It was sequenced on a PacBio SQL2. Compared to it, the sequencing of the PacBio ultra-low input DNA library was very, very successful. And on average, the sequencing output was about 470 gigabases. In total, we made 2 smart cell sequencing runs for the ultra-low input library. Comparing the 3 different genomes from Elysia timida, Elysia chlorotica, and Placobranchus ocellatus showed that the contigent 50 from Elysia timida was highest with over 1 megabases, compared to 30 kilobases for Elysia chlorotica and 5.5 kilobases for Placobranchus ocellatus. The scaffolding 50 of Elysia timida was a little bit lower compared to Placobranchus ocellatus. But again, the complete bascule value for Elysia timida was highest with over 95%. So what can we see? The Placobranchus ocellatus and the Elysia chlorotica genomes are still highly fragmented. And this is probably because only short-read data were used for the genome assemblies. And we could show that the ultra-low DNA LibPrep was very, very efficient for long-read sequencing compared to the PacBio low DNA LibPrep. Outlook: we already have dovetail omni-seq data available for Elysia timida, and we would like to improve with these data the genome assembly. Further, we would like to do an annotation with transcriptomic data. And with this, I would like to thank you for your attention. And if you have any questions, just come to my poster.